I want to take this opportunity at the beginning to thank Krell Institute and CSGF for putting together such an awesome program and for allowing us to expand our horizons and to pursue our passions. So my particular passion is protein folding. And I was drawn to it because proteins are the nanomachines that work inside our cells that essentially make life possible. So everything that effectively takes work inside your cell and as work between and communication between cells, it's all done by proteins. So in that sense, it's not just what makes life possible, but proteins acting all together and coherently essentially is the life force that philosophers and scientists have been trying to understand for a long time. A lot of my PhD involved looking at the complicated structures of proteins because they do all these different functions in our cells. Now, proteins are actually linear chains composed of an alphabet of 20 different types of amino acids. And the sequence along this chain on the protein is determined by the sequence in our DNA. But once the sequence on the chain of the proteins is determined, the protein then folds into its three-dimensional structure based on what the sequence is. And the three-dimensional structure is then what gives this protein its function, both mechanically and chemically. Even though I spent a lot of my time looking at the overall structure of proteins, and there are so many different types of structures of proteins responsible for all sorts of different functions, this talk I'll be focusing on something in particular that unites proteins. So if you were to look at this pie chart of all the proteins, the green section of the chart denotes proteins that have a local type of structure called a secondary structure called the beta strand, which is shown as green arrows here. The red part of the chart are all those proteins that have another type of local fundamental structure called the alpha helix, shown here. And the yellow are all those proteins that have both. So if you were to look at this, even though there are so many different types of protein structures, and in fact, I have to give them the structure classes with names like jelly row and all the like, if you were to take a step back, you see that proteins are actually united by fundamental structures. So this talk, I'll be focusing on the alpha helix, which 75% of all proteins possess. So what is the alpha helix? If you were to look at this cartoon rendering shown in the previous slide, it's actually the helix winds along the backbone chain of the protein, and it's stabilized by hydrogen bonding between every fifth amino acid along the protein. And the alpha helix thermodynamically has been well understood since the 50s. In fact, you can just directly cross-apply the one-dimensional Ising model for spin systems that were developed to study those physical systems and just cross-apply that to the helix to coil transition. And you're able to predict melting curves, et cetera, et cetera. But what's not so clearly understood is the kinetic picture. How fast does this helix form? And what is the mechanism? Is it diffusion limited, or does it have to cross some sort of barrier in order to form this helix? And in fact, experiments on how quickly these helices form range from sub-nanosecond to microseconds. So 4 to 5 orders of magnitude in the range of time scales that people have been able to measure for the formation of this. So that's kind of a big range. And we want to understand, well, what contributes to this range and what are exactly the fundamental processes. One thing that contributes to this range is that alpha helices come in various lengths. So if you have a longer helix, obviously it's going to take you a longer time to propagate this helix and make it bigger. So what we want to understand is what are the fundamental time scales for the fundamental events, that is the formation of the very first turn of the helix, and then how long does it take to propagate each turn of the helix. Those fundamental events should be about the same for all proteins, and that should unite this huge time scale into those constituent events. So in order to do this, we focused on the formation of the very first turn of the helix. And in order to do this, in both a computational and experimental effort, we designed a pentapeptide composed of only 5 amino acids. The advantage of this is that whatever we measure in an experiment is only the formation of that very first turn of the helix. And unlike previous experiments, is not convoluted by initiation of helix along the chain and also propagation and unpropagation of the helix. So whatever you measure here is the fundamental event. There are 2 different sequences that we designed basically mutations of each other. We want to look at what is the effect of mutations even at this very smallest scale of proteins. So one of the sequences, which we call WH5, has the three middle sequence amino acids are alanines, which means that their side chains, the chemical that bonds to them here is just a methyl group, so carbon, 3 hydrogens. So it's bulkier. The WGH5 is exactly the same, except that the 3 amino acids in the middle are glycines. So instead of having a methyl group, they have just a hydrogen atom. So there's less bulk there. And because there's less bulk here, unlike WH5, WGH5 can form both the left-handed and the right-handed helix. That is both the corkscrew counterclockwise and clockwise. Whereas because of the bulkier side chain, WH5, like basically all proteins found in nature, which have side chains that are bulkier, can only form the right-handed helix. So first, we developed a coarse-grained analytic model to be able to predict the order of magnitude timescale that this event should happen. So we want to go from 4 to 5 orders of magnitude to within like a factor of 2, just to see if we can understand how quickly this event should happen based on the physics that we think dominate this type of event. So if we were to look at this pentapeptide, this protein with just 5 amino acids, essentially, it's a very complicated process, and what makes protein folding so difficult to predict is that there's so many degrees of freedom, which add exponentially to the complexity, right? Even in this small protein, there are many degrees of freedoms of wiggling. Essentially, we want to predict, well, if there are all these different degrees of freedom to wiggle, how long does it take for all of them to wiggle so that they're in the right conformation so that the hydrogen bond can happen and snap the helix into place? In the model, there are effectively 3 key degrees of freedom denoted by psi 1, psi 2, psi 3, which can rotate these torsional angles. And so we modeled this conformational diffusion as the conformational diffusion of these 3 torsional angles, and as such as a superposition of these 3 modes of torsional diffusion. So there's a superposition of these types of motions. Now, if we were to look at just one part here, the degrees of freedom. So rotation around this bond is called psi, and rotation across this bond is called phi. Now, phi is not drawn, labeled here for the following reason. If we're to look at what's called a Ramachandran diagram, so we're plotting phi, rotation along this bond, and psi, rotation along the bond drawn. The white regions denote those phi psi, those areas in the phi psi space that are sterically prohibited. That is, there's going to be a clashing of atoms, and so they're prohibited. The dark blue regions correspond to the regions that are allowed, that have no steric clashes. And then you see these light blue regions corresponds to the region where there's a slight steric hindrance, and that the 2 atoms are very close together, but not so close that their centers are going to overlap with each other. And what's interesting is that the Ramachandran diagram conveniently decomposes the conformational space into 3 regions. And the 2 regions I want to focus on is this dark blue region contains a smaller subpart, this light yellow region, which is the beta strand. So that means if all of these conformations are here, so if this set of conformations, this set, et cetera, are all in this yellow, it'll form that beta strand structure I showed you earlier, one of the 2 types of secondary structures. On the other hand, if all of the degrees of freedom lie in this region, this little red region inside this blue basin, then the structure will form an alpha helix along the chain. So the model basically says, well, how long does it take if all the degrees of freedom are outside? How long does it take for all of them to diffuse and all come into this region? So this degree of freedom, these degrees of freedom, these degrees of freedom. And what we did was we separated a time scale so that we looked at the fact that there is this barrier, this weak barrier between the 2 large blue regions, which are the basins. And we said, well, the time-limiting step must be the time it takes for these degrees of freedom to all diffuse into this overall basin. And once they're in that basin, before they can escape from this weak barrier, they will all come quickly into this red area. So by decomposing this into a 2 timescale problem, we can then discretize the problem and solve it analytically. And so the time it takes to form this first term of the helix is tau divided by p cubed. Tau is the time it takes for each of these degrees of freedom to forget its conformation. So forget where it is. That's the memory loss. And p is the probability that once it's randomly scrambled, it's in the correct basin. So allowing for this, we can then solve for what is the expected relaxation of each of the degrees of freedom. And we know from translational invariant symmetry that it must have an exponential decay in its memory with itself. And then relating that for very short times to the Einstein equation calculated from the fact that this is following Langevin dynamics, that is dynamics with friction as well as random force inside this water medium. We can then relate these two still for short times and divide out the time dependence t and just get the relation what tau is. We can calculate what p is because p is about 1/2. That is half of the time it'll be in this region, half the time it'll be in this region. So we have this analytic expression based on only the viscosity, the volumes of these masses, Boltzmann's constant, and the temperature. Okay, so how well does this predict how quickly the helix can form? So here is the prediction for it. So this blue line shows what it is, the predicted times if we assume just the bare volumes of the atoms in the protein. And the dotted is the more realistic assumption that we had one layer of water because the protein usually carries a hydration layer to it. And the red are the experiment. Note that what this does is supposed to predict within like a factor of 2 because we make assumptions like things are spherical, et cetera. So whatever, it's not fitted, and even though it looks like the experiment lies on the prediction, that's just a coincidence. It should be right within like a factor of 2, but we don't expect it to be exactly right. But this just says that alpha helix initiation should happen on a few nanoseconds, okay? But in order to get the more fine detail, we did computational molecular dynamic simulations on an explicit box of water for the structures that we studied. And we were able to get hundreds of independent folding events so that we can actually tabulate the statistics, not just a single event, but actually get enough statistics to actually simulate the actual signal to noise of an actual experiment. Okay, so here's what we got. So for WH5, we actually got that if we were to plot the histogram of folding time versus the frequency of that folding time, and to fit it, we can only fit it with the 2 time scales. It does not fit with 1 time scale, and this is consistent with the experiment that we performed. And the time scales also agree. Conversely, for WGH5, that is the one with the less bulky side chain, the 1 and 2 time scale fit gives the approximately indistinguishable curves, and likewise, the experiment gives only a 1 time scale fit. Okay, so we see that we do have an agreement, and we can understand then what's going on in the experiment, but also we can explain why we have 2 and 1 time scale. So this goes back to that assumption we made in the model way back here, and that once the degrees of freedom can diffuse into the correct basin, which is a time-limiting step, they can then quickly anneal into their conformation. So what this is saying is that in the bulky side chain, where this barrier is present, the slow time scale is the time it takes for these degrees of freedom to anneal into the correct basin, and the fast time scale is the time scale it takes once they're in that basin to anneal into the correct alpha helix shape, so that they do not escape out that weak barrier. And this is borne out by the simulations, and in fact, if we were to plot the free energy landscape from our simulations, so the way that this landscape is parameterized is that the radial coordinate is the distance between this oxygen and this nitrogen. So when they're the correct distance of a few angstroms, that is when the hydrogen bond is formed. The angular coordinate in this polar graph is the twist of the molecule. Okay, so just like I said before, you see that for WGH5, which has the small side chains, you have 2 of these minima corresponding to the left and the right-handed helix. They're equidistant from the center because their bond distance is the same, right? They're short bond, but they are on opposite sides because they have opposite chiralities, okay? And likewise, for WH5, which has the bulkier side chains, you only have 1 minimum because the other minimum is sterically forbidden. But more importantly, by looking at the free energy landscape, which we get from our simulations, we can actually go back and validate the assumption that we made in the model, which that it is indeed, you can separate it into 2 time scales. So in the slow time scale is the time it takes for it to diffuse from the large unfolded state to cross into this basin here, which is separated by the rest by a low wall in the energy landscape. But the fast time scale is that once it's in there, it'll quickly diffuse to here. So this not only explains what we see in the experiment, but validates the model, analytic model used. So to summarize, the speed limit of protein folding is 1.5 nanoseconds. This is the fastest that any structure can form in a protein. It's diffusion limited, and the energy landscape and the folding is dictated by the sequence. That is, if you make a mutation, you'll affect not only the equilibrium structure, but actually the way that the path it takes to fold, even on the smallest scale. And this is an example where there's a marriage of both theoretical, computational, and experimental efforts to get a complete and reliable picture of what's going on. So with that, I would like to thank my advisor, and I'd like to thank the DOE again.